Good morning, or good afternoon if you're in the East, and welcome. I'm going to be talking to you today about a subject that's very close to my heart, and I hope it's close to yours. And the title of the presentation, I think, says it all. What will it take to overcome the dismal success rate of cancer biomarkers and molecular diagnostics? And those two are obviously very closely related. I want to remind you that this presentation is CME eligible. So if you want to click that for your CME credit, you can do that now. The story is a really interesting one of biomarkers, and I'm going to take you through that story. A little bit about myself first, though. I'm the co-director here of Complex Adaptive Systems, and I'm the director of something called the National Biomarker Development Alliance that was developed over the last 2 to 3 years. I'm also a professor of the School of Life Sciences at Arizona State University, and formerly the deputy director of the National Cancer Institute before I came to Arizona State University. So if we start now to talk about the issues today, I'm going to actually talk to you a little bit about biomarkers and why biomarkers are the foundation for personalized medicine and often called precision medicine, and a little bit on what they are and the state of the field. And then we're going to talk about some of the problems in the field. And the reproducibility of biomarker discoveries is a major problem. However, I'm going to talk to you a great deal today about the identification of the problems in the field and some of the solutions that we have come to. And I'm hoping that I can convince you today that although we have real problems, I think we have the greatest opportunity that I've seen in the history of medicine to actually change what's going on in this field and change the direction of medicine for patients. And that's going to be a great part of our discussion today. So let's get right into it. So biomarkers are a rational foundation for personalized medicine, and why is that? Here's the reality of cancer, and we think about these statistics a lot, and we talk about them a lot, but they're so mind-numbing sometimes that we kind of forget that it's something that is actually ever increasing and increasingly an economic disaster in terms of healthcare. So if you look at these numbers year over year, the number of new cases in the US in 2013 was 1.6 million. I used to talk about that number being 1.4 million. In the future, we're going to look at increases of upwards to 40% in terms of cancer, especially obviously in people over the age of 55. As 77% of cancer is diagnosed in individuals over the age of 55. The good news is we have a lot of survivors today, and that population is going to also continue to increase, and some of the morbidities in those patients will become bigger problems as we go through the next few decades. The problem with cancer, essentially, is that we really haven't done much about the mortality rate over the last 40 years. It's incrementally a little better, but we have a long way to go. If you think about the economic consequences of cancer, they're really daunting, because if you look at a 30 to 40% increase, and although the cost of cancer is variously reported, it's certainly over $200 billion a year, and is moving upwards to probably approach at some level, at some state, the $1 trillion level. So if you look at that in the context of molecular sciences, going back several years, almost 50 years, to the discovery of the Philadelphia chromosome, there is a story that actually has driven this ideal of personalized medicine, and it's actually founded, or it started all those years ago with the discovery of the Philadelphia chromosome. And this is a chromosome that has a translocation in the BCR gene and the ABL gene, and that fusion gene turns out to be the cause, certainly the major factor in the causative pathway for chronic myelogenous leukemia. And so in 2002, we approved a drug called Gleevec for this particular disease. And at that stage in 2002, this particular drug was curative for most of the patients that actually received it. And that's true to this day. We've learned a lot since then about how this particular gene can continue to mutate and change. So resistance develops in these patients. But this story actually started us on a road to personalized medicine. And there have been many stories since, not enough, but a few. We had a recent story, for example, with malignant melanoma, where we're seeing BRAF, that particular gene, be very, very diagnostic in the causal pathway potentially for some of the melanoma that we see. So this was not a bad basis to start thinking about precision medicine. And if you then think about a little further about healthcare, the realities going back to the cancer burden that we are anticipating in the next 15 to 20 years. By the way, this isn't just true for the US, this is a global issue. And globally, it's quite profound in terms of the numbers of cancer patients. But if you look at healthcare spending, going back to that number of cancer patients that we anticipate, in 2013, it was about somewhere around $3 trillion, and we expect that to increase by 4% or more per year for the foreseeable future. So it looks as though this particular idea of healthcare spending increasing is going to continue to be a fact of life. And I was on a panel the other day in Washington where a very prominent payer was able to predict, and in a very scary fashion, I might add, that currently many cancer patients will have to spend half their income on treating their disease, and in the future it could be 100% of their income. So these are very frightening statistics. And so 21st century medicine, early detection, disease prevention, targeted diagnostics, targeted therapeutics is the way that we see the future. And if you believe that, which I hope you do, then biomarkers, those molecular changes that actually are diagnostic of the disease often, are going to be the key to the future of personalized medicine. So it's all about biomarkers, and you can see that in the numbers of meetings we hold, the numbers of conferences that are held, the number of publications. Biomarkers are a very big deal. So their potential can almost not be estimated at this point. It's probably the highest value proposition in personalized medicine today. And to realize this value, they're going to have to be developed for therapy development, for example, markers of efficacy already in use in terms of companion diagnostics, early detection. This is a very important area that isn't going all that well for cancer yet because the molecular diagnostics industry doesn't have a great business model at this point. Choosing treatments or stratifying patients is already well underway. And we have enabled some of that through whole genome sequencing, projects like the Cancer Genome Atlas, which Francis Collins and I started when I was at NCI, has paid off in many ways to begin to stratify these patients based on their molecular profiles. Assessment of treatment, prognosis, or essentially determining what the clinical outcome is going to be by using biomarkers, or predicting response to therapy. Those are not the same thing. Prediction in terms of surveillance, in terms of the whole of population surveillance is very, very promising. And the whole issue of surrogate endpoints for clinical testing. Surrogate endpoints we're going to talk a little bit about in a minute, but this is a very important concept and one that needs a lot of attention from our communities. So the value proposition of biomarkers and their relationship to the achievement of personalized medicine is an absolute. So a few definitions, and then we're going to talk a little bit about the state of the field, but this is a confusing field in a lot of ways. I held a meeting here about 18 months ago with some of the smartest biomarker people I know, and it took us the whole morning to come to an agreement on a definition for a biomarker. However, as in all things, there is an official definition for a biomarker, but there's a lot of confusion around this definition. A characteristic, biological or physical, and it can be an image. Blood pressure is a biomarker. So it's not just about molecular changes. That is objectively measured and evaluated, usually a change, as an indication of normal biological processes, pathogenic processes, or pharmacologic responses to a therapeutic intervention. In other words, you can use a biomarker to measure a change once you give someone a drug. Ideally, this is very important, it should be clinically relevant, fit for purpose. That's an FDA term that says you have to know the context of use for your biomarker if you're going to submit it for regulatory review. It has to be measurable, reproducible, quantifiable, and directly reflect the current state, whether it's normal or altered. That's a lot. Those are a lot of parameters, and they're all very important. A surrogate endpoint, and sometimes this is called a surrogate biomarker, is a laboratory measurement or physical sign that is used in therapeutic trials as a substitute for a clinically meaningful endpoint that is a direct measure of how a patient feels, functions, or survives, and that is expected to predict the effect of the therapy. Bob Temple said that in 1995 from FDA, and that's still in effect. So a biomarker that substitutes for a clinical endpoint is a surrogate endpoint. And you have to remember that just measuring a molecular parameter may not tell you how a patient will feel, function, or survive. So as a consequence of that, we do not have many surrogate endpoints in the cancer field. Cardiovascular disease has quite a few of those. There are not many surrogate endpoints, period, because it's extraordinarily difficult to find biological molecular changes that are in the causal pathway of the disease. And it's also very, very difficult to do the trials to relate it back to how a patient feels, functions, or survives. A lot of biomarkers fail because of that problem. So a couple of other terms that you need to know. Obviously, to measure a biomarker, you have to be extraordinarily careful in building an assay that is sensitive and has specificity. So the extent to which an assay correctly identifies the disease, when the disease is really there, is a true positive. Specificity is the extent to which an assay identifies true negatives when the disease is truly absent. And that's in the presence of all confounding factors. Precision is the closeness of agreement between independent test results once you obtain your assay under defined conditions. Fit for purpose is an FDA term that's used in their biomarker qualification program, which defines the context of use. Assay development specific for stage of the drug development considers the intended clinical use and defines the regulatory requirements. So this is a very, very defined pathway now. And then just those of you that aren't in the assay development business, you have to know how your assay is performing. Normal populations are generally never totally normal, and disease populations are never exactly homogeneous. And so you have to perform a lot of operations and a lot of tests to determine what the overlap is between those populations, and that's called the receiver operating curve. It's been around for decades. But assays don't generally specifically distinguish disease from normal with 100% accuracy. So this is the way you actually come to analytical validation. Analytical validation is what defines a molecular diagnostic versus a biomarker. So a lot of terms, and there's a lot of confusion in the field about these terms, and not easily understood. So as a consequence, it's produced a lot of problems for us in the field. So just so we're on the same page, biomarkers essentially that we're talking about today, we're talking about molecular biomarkers today. We're not talking about imaging or some of the other things that we could be talking about, but we're talking about changes in the genome and or the transcriptome or the proteome, microRNAs. It's this whole range of molecular changes that can occur. So proteomic information, genomic information to help a physician make a clinical decision about a patient care. That's increasing. They exist generally in the literature and clinical trials at various stages of validation. And you can read about a biomarker in the literature and not really understand the extent to which it's been either analytically validated or clinically validated. And you can now qualify these kinds of things, these biomarkers, with the FDA for a specific context of use. And that you can go online to FDA's site and see what the qualification programs look like. Molecular diagnostics use this information to distinguish between normal and diseased tissues and to help a clinician make a decision. These molecular diagnostics are analytically validated and clinically validated, and they receive approval from FDA on a couple of pathways through a PMA or a 510K, and I'll come back to that in just a second. And these agents, when they become molecular diagnostics, they may be used as diagnostics and or companion diagnostics for drugs. So just so we are all on the same page in terms of FDA here, so if you have a biomarker that you believe is on the road to becoming molecular diagnostic, then there's a lot of work that you have to do. So PMAs are pre-market applications, and they actually involve a lot of data and a lot of steps and a lot of money. So molecular biomarkers to be used as companion diagnostics or molecular diagnostics are categorized as class 3 high-risk devices. And so you need the support of data through an IDE, essentially an investigation device exemption trial, to show that your biomarker is going to do what you say it's going to do, it's analytically validated, and it's clinically validated. A 510K is an easier route through the FDA because there's an approved device or a predicate device, and you still have to collect the data through an IDE trial. So these are very, very similar pathways. The PMA application is the entry pathway for a device, for example, now for a molecular diagnostic that's going to be used in the clinic quite broadly, considered a high-risk device. And then in July of this year, FDA has indicated that it's going to regulate laboratory-developed tests. Historically, it has not regulated these tests. It's going to start its regulation on a risk basis, essentially with those tests that are already in the marketplace that are done under the CLIA regulations, essentially, which have nothing to do with the FDA, by the way. So they're going to start with those tests that are considered high risk, and over the next nearly a decade, they're going to phase in other laboratory-developed tests. And you can read those new regulations in the Federal Register, and the draft guidance is out, which means you can comment on this. And there is everybody is in uniform agreement about this, and you have a chance to comment on it. So, so to take you a little bit through the state of the biomarker science or the biomarker field, I want to just do a little summary for you so you know where we are. So do we have a lot of biomarkers? Yes, we have a lot of biomarkers. We're awash in biomarkers. My colleague here, George Post, who's the co-director of Complex Adaptive Systems with me here at ASU, published a paper in Nature all the way back in 2011. This has gotten bigger. That balloon has gotten bigger, actually. But he estimated just the number of papers documenting thousands of biomarkers was over 150,000. We normally use less than 100 biomarkers in the clinics. That will give you some idea of the state of the science in terms of what gets approved. One of the areas, obviously, that we have bet very heavily on in the molecular sciences is proteins. Those are the molecules that do the work of the cell. Lots of these have been submitted in one guise or another to the FDA, but there's been less than one approved per year since the early 1990s. So there are lots of bias in these studies. There are no standards that are being adhered to on a national basis. Reproducibility across the labs is a problem, et cetera, et cetera. So as a consequence, this has hit the molecular diagnostics industry pretty hard in terms of getting technologies that are reproducible, robust, and can withstand the scrutiny of regulatory review. So as a consequence of this, I mean, proteins are not being approved. we don't have that many diagnostics approved at all yet in terms of cancer. But one thing we are seeing and have been seeing for decades is in cancer, especially where too many of our clinical trials are failing. And that has to do in many ways with the biomarker space and biomarkers not being appropriately either qualified or analytically validated. And when you get into late phase clinical trials, we're having a lot of attrition, long duration trials, and very high cost. And if you look forward in this space in terms of pharmaceutical development, it's going to get worse, not better. As we bifurcate these diseases into more and more subtypes, this is going to be a really interesting problem that the pharmaceutical industry is facing. The cost of a PMA, in case you want to get in the diagnostics business, it's pretty expensive. And it involves several areas. You have to know, first of all, you have to discover the biomarker, then you have to essentially do some early development on it. And then there is the process of getting an IDE, essentially getting a device trial set up, and you have to do clinical safety testing, then you have to do a pivotal clinical study, and then you have to apply for your PMA. And all of that costs money, and it's somewhere between $94 and $100 million, which essentially this is pretty equivalent to what we see with filing an IND for a drug. And obviously, this makes it very difficult to get into this field, especially if you don't have a pretty good idea that your biomarker is going to succeed as a molecular diagnostic. So recently, we've seen a lot of this kind of data and these kinds of questions on the cover of all kinds of journals and magazines. Now, I should tell you this issue's been on the cover of Science, it's been on the cover of Nature. However, scientifically, we know we've got a problem when it's on the cover of The Economist or it's on the cover of The New Yorker. So this issue of reproducibility in research has become a national conversation. And a lot of the reproducibility issues are in the biomarker space. And I'll come back to that. This is just the Science, Nature, and PLOS covers on this kind of thing. So, we do have a problem. We're going to talk a little bit more about that and what it means. So how widespread is the problem? I just picked off a few of these, but you can go look in the literature and you can go look at papers, retracted, et cetera. But so this is just a few of these. I just looked at some of the molecular epi studies. For example, 5 of the 7 largest studies didn't classify patients better than chance. And microarray signatures, I know about this study essentially to predict patient response. It was named one of the breakthroughs actually in 2006. And in 2009, it was shown to be wrong in terms of labeling and due to labeling and indexing errors in the laboratories. But these are the 2 studies that have gotten the most attention and been published on. Essentially is the Amgen study, where Amgen scientists attempted to reproduce 53 of the seminal publications in the field that they were interested in, which is drug development. And these were discoveries that were purported to be great targets, essentially. They could reproduce only 11% of those, and Bayer had a similar experience with a rate of 25%. So this is this reflects. This is diagnostic, no pun intended, of the field, and I would like to challenge you to believe that it's also one of the biggest opportunities we have, and I'll tell you why. So this is impacting our ability to deliver the discoveries that are so important to patients. And so I think these are all fixable problems. And so when I came to ASU, coming up on 2 years ago or so, I decided when I was at NCI, we worked closely with FDA to see if we could develop, what is the trajectory for a biomarker? What do you need to do to get it from a discovery all the way into patients? We didn't get very far with that problem. We worked on other problems, but this is a very sticky one. So when I came to ASU, I decided that we would tackle this problem because this is not a problem for the government, for the industry, for advocacy groups, for universities. It's a problem for all of us. And so what we decided to do is see if we could bring these sectors together to begin to discuss the problem, identify the problem, and figure out what the solutions might be. So that's the story that I want to tell you. So we set up something called the National Biomarker Development Alliance, and essentially it's a trans-sector alliance that's assembling people who want to change this, really want to change this, organizations and stakeholders to develop a deep understanding of what the problems are, and I would argue that most of us didn't know what they were when we started, and also to create some solutions that are implementable and will actually change this and bring these biomarkers through to success and also really change the lives of patients. So we started, it's about 18 months ago or so, a little more than that, holding workshops around just starting to ask ourselves questions about how do we discover biomarkers, and then how do we actually develop them? Do we have a system to develop them? And ending in we had a last one in July talking about, what are the clinical trials and what are the real challenges in clinical trials for biomarkers. And we worked our way through a whole range of problems and issues that I'll tell you a little bit about. So this is what we've learned. And so in a very few slides, I'm going to tell you almost 2 years of work of due diligence on understanding what the problems really are. So I can show you here what the problems really are in this graphic, because this is the graphic that we've come to. One, this is an end-to-end process. Developing a biomarker is an end-to-end process. You start with discovery and you end with biomarker validation in a clinical trial, be it for as a companion diagnostic or as a diagnostic per se. Each of these areas up here essentially these represent real decision points in here where you have to make a decision to move a biomarker, not unlike a drug, from one of these areas to another. And we distinguish early discovery from translatable or later discovery from assay development, although these clearly overlap, and this overlaps, but they are quite distinguishable sorts of modules in this whole process. The qualification process and then the validation process. We are really concerned over here because so many things are leaving early discovery that should not leave early discovery. Just as we need to kill drugs more quickly, we need to kill biomarkers more quickly. So this is a graphic representation of what I believe an end-to-end process should look like. And if you look at this, we need standards at every one of these decision points. We need standards even starting over here. Now I should tell you that what I'm about to tell you is, I think, the opportunity of a lifetime in terms of cancer research. I think that we have over decades been making fabulous discoveries. We now have the opportunity to take some of those discoveries, not all, but some, and in the future maybe more, through processes that will more quickly get these discoveries to patients. And I think that's the opportunity that will actually totally erase any issues of reproducibility and concerns for reproducibility, because we all know as scientists that in early discovery there are discoveries that will be very difficult, if not impossible, to reproduce. And that's okay, because discovery is discovery in many cases. But if you want your discovery to end up in a patient, there are things that you have to do here to get to here. And one of those things we have discovered is standards. And we are not good in our communities at standards, but we're going to have to become very good at standards. So to go from here to here, this is one of the things that we discovered. Absolutely, we're going to need standards. So we pursued each of these modules and tried to determine what it was in each of those modules that made biomarkers fail. And what we came to is the two big culprits here are in that discovery and translatable discovery realm. And if you think about, and this is due to talk to a lot of scientists, and every scientist will tell you, almost to a scientist, that the biggest barrier that we have in biomarkers is the complexity of the science. And everyone agrees on that. The complexity of the science is what it is. And we're going to have to understand that as we deconvolute the complexity, we're going to have to bring rigor beyond understanding whatever we've deconvoluted in terms of complexity. We have to take that piece of information and turn it into knowledge. But this is the most cited barrier by the scientists in the field, actually. And if you look at why that's true, and those of you that work in the genomics field know this, we learned this in the Cancer Genome Atlas, absolutely, is that the landscape of genomic alterations in cancer is overwhelming. And if you look at this slide, you'll see that these are mutations in individual non-small cell lung cancer, and these are the drug targets. And I can tell you from doing 500 samples in every TCGA tumor, every individual has some slight variations. You can group these into subtypes based on their molecular variations, but in fact, this is very concerning in terms of the development of new drugs. But this reflects the complexity of the disease that we're talking about. And then if you go to the protein level, again, supporting my colleagues on the complexity question, this is 500 biomarkers, and this is the connections, essentially, conditionally dependent connections in glioblastoma. And this is a tumor that we do very little about in terms of therapy, as you know, but the complexity is reflecting the networks that actually are communicating with each other at the protein level, which is extraordinarily complex. And then if you take this to the more popular level that we're dealing with today, which is pathways, and we have discerned a select number of pathways, and we like those pathways. It's very, very convenient to think about pathways, but pathways are convenient models. They are no more than that, and it's not that simple as all these pathways are interacting in real time. So just supporting our colleagues, the biology is extraordinarily complex. But let's look beyond the biology and say that we have those hundreds of thousands of biomarkers every single year. Is it a solvable problem? And I would argue that it absolutely is. We made a really important discovery that I think could change the field, and I hope, when I say it's a surprising discovery, it shouldn't be surprising to any of us, but what we found is the following. The problems in biomarker discovery usually begin in early discovery. So essentially, it makes them impossible to reproduce often, and the big killer here is starting with an irrelevant clinical question. In other words, a lot of our biomarker discovery doesn't start with a relevant clinical question, and we back into a question. You know, we discover something and then we go looking for a rationale. That's not the way to do something you want to put into patients. So biomarkers are generally based on convenience samples, unfortunate, but true. They have a lot of people who discover biomarkers don't know much about statistical design. We don't have technology standards generally that are applied universally. We know little sometimes about the provenance of our data, and the data management aspects of this are critical. And the analysis and the analytics are often inappropriate or inadequate. So all that adds up to this. These are the key barriers. In these six, I want to call these strategic elements of biomarker discovery, also lies the solution. So the number one, I think, most important aspect of this is probably the lack of a relevant biomarker-appropriate clinical question. And that means we have to very carefully assess biomarkers relative to what they're going to be used for by a clinician for making a decision about a patient. Inadequate experimental design, either in terms of the statistical design, whether or not it's actually going to address the clinical question. Poor quality biospecimens. This is the bane of at least my existence, and I hope it will become yours, because we know how to solve this problem. but many of our samples are inadequate. Sometimes they're, our experimental designs are sampling, from the wrong populations, et cetera. Lack of or disregard for tech standards and poor quality data, and inadequate analytics and analysis. And these we call kind of the big six. If you go into the literature and you read a paper on biomarkers and you can't put a check mark by each of these, then you likely do not have a biomarker. So just a couple of words here about this in terms of looking at a couple of these. So this is essentially the HER2 test. And if you look at the differences here in IHC and FISH in breast cancer, there is a loss of signal with time to fixation. And there is a gain of signal on the right-hand side, essentially in colon cancer. That's a big deal. And if you look at the statement from Invivimed, without knowledge about tissue processing methods and assurance of rapid tissue fixation, protein expression, data are unreliable, and understanding of pathway activity is not possible. That's a very damning kind of statement. And if you look at, sorry, I'm going to go back, and now I'm going to go forward. If you look then too at the entire landscape of devices that's coming along now, every one of them will have different requirements in terms of those 6 parameters that I just mentioned. And so our goal is to raise the bar for biospecimen quality for all of these areas, and we're doing something about that. Bias is a big deal in all of these studies and experimental design. As good as you think you are, specimen collection is another area where is one of the key areas actually where bias really makes a difference. And then there's multiple observations. Don Berry wrote this paper in 2012. It's very relevant in the sense that when you're making recordings of observations over time and multiplicities are rampant in the work that we do, the chances of false positives become very high. So all this leads me to a conversation that we had with FDA, and they've been very, very happy in terms of working with us on the National Biomarker Development Alliance. So Janet Woodcock, who is the deputy director there for drugs and probably one of the most innovative regulators on the planet, if not the most, she made a statement at our first meeting of the NBDA where she said it is not FDA's job to develop the standards for biomarker development, it's the job of the affected communities. And that's true. So what we have done is to pick up on what she said all the way back in 2002 or 3 in something called the Critical Path Initiative, which you can find on FDA's website, is that FDA realizes that essentially biomarker development and streamlining clinical trials are the 2 most aspects of really improving medical product development in this century. So FDA agrees with us, and they have told us that they're not going to develop these standards and they're not going to develop these processes. It's our job. So the NBDA mission is to collaboratively create standards, meaning best practices, guidelines, standard operating procedures, and we're not going to reinvent wheels. If there are good guidelines out there, then we're going to use them. For a systems-based end-to-end biomarker development process or processes, and in doing so, advance precision medicine. That statement means a lot, and it says a lot. And I believe it can be done, and I believe having a system that is end-to-end so that an investigator has some direction when you start to develop a biomarker will be invaluable. So coming back to this, we have, I think we have, I've told you enough about this essentially that you understand it's an end-to-end process. These are all decision points. This is critical. Once you move into this phase, all kinds of things start to happen in terms of the big 6 that we talked about. If the big 6 weren't considered over here, the chances of failure over here become monumental. So I repeat, right clinical question, right experimental design, great samples, technology standards, great data and great analytics will take you to the finish line. So these problems are all solvable. Other fields have faced problems like this successfully, and I think we can as well, and I think we will. And changing the whole field of cancer research without compromising innovation and discovery or early discovery is going to be critical to actually achieving precision and personalized medicine for patients. And I think we have to deliver on this promise. We have to. Not because we're all dependent on, you know, the government for grants or our institutions for salary increases because we publish a lot. It's because we can actually save the lives of patients, and we can prevent this disease. We can detect it early if we solve these problems. And that will change. That won't just change cancer. That will change every other chronic disease, actually every other disease. I talk all the time to people in the psychiatric disease field or in, you know, in Alzheimer's, et cetera. They have exactly the same problems we have. So the problems of biomarkers are pretty rampant. So we're doing some things now to start to bring solutions to the table. We're starting to develop this idea of can we actually talk about surrogate endpoints and begin to define it in a new way. The chances of actually us being able to come up with single changes that are going to be absolutely aligned with the clinical endpoints that FDA recognizes are increasingly unlikely. Maybe out there's going to be a way to look at the natural history of the disease and redefine surrogate endpoints. We're holding a workshop on this in December in Washington. So the other thing is we're trying to set up a commons for biomarker designs and clinical trials protocols. So the best of the best will be available to everybody. And we're holding something called convergence conferences beginning this December to talk about what do we have to do for biospecimen standards to get reproducibility, not just in genomics and proteomics, but in other aspects of molecular markers as well. And we want to partner with the College of American Pathology to bring this back to hospitals so that it will impact patients. And then we'd like you to join us in this. We hope that you will join us and become part of the solution. And you can go online and see us at www.nbdabiomarkers.org. And your institution is welcome to join us. We haven't started our membership drive, but it'll be starting in about a week. And we're in Scottsdale, Arizona, but we are actually virtual. We have probably about 50 scientists working with us and a lot more joining us. So I want to have time for questions, so I'm going to stop there. But just to give you an idea that we're working, things are happening. This is the press club in January where we launched the NBDA. People like Janet Woodcock was there and a lot of other people. And these are some pictures of our workshops. When I say workshops, people are really working. They don't come to listen to, you know, endless PowerPoint presentations like this one and then go home, or more likely work on their computer. But they work. We actually work together to try to come up with solutions and to, first of all, identify the real problem and then come up with solutions. So we are underway. We think that you know, we're going to actually bring some new thinking to the whole biomarker space. It is the most important action I think any of us can take in terms of enabling this whole concept, and it's still a concept in many ways, of precision or personalized medicine. But there's much work to do, and if you go online and you look at the NBDA website, you'll see, and you'll be able to access actually many of the reports that are up there, a lot of the things that I told you today. There is every opportunity here for us to reshape this entire field, and it is the job of the stakeholders. It's our job. So I'm going to stop there, and happy to talk more about biomarkers. We have some questions, and I'm going to take some. We actually have a lot of questions. So I'm going to actually take them from the top. And so some of them are comments, but so somebody was very nice and said that it was an excellent talk. Thank you for that. The details are very important to achieving the final goal. Let me see, here we go. Okay. How do you see the tools of the field defining the level of deep research that appears to be driven by other factors at this time, with advancements overlooking the finer details gained by detailed investigation? So you're absolutely right, and this question's coming from Deborah at DKT Lab Consulting. So you've asked the question that actually I think is the reason that we find ourselves where we are. And that question essentially is we've not ever taken the time to figure out what are those details that will take us to success. We're just in this frenetic period of doing discovery and discovery and discovery. Some of that has to do with not being able to actually understand the field in some ways, or in many ways, or the biology certainly, but others, some of it has to do with just our unwillingness to accept the fact that the details matter. And you're also absolutely right about the fact that as we make these advancements, you would overlook detail at your peril. There's no doubt about that. So, second question. So when I say it's not that simple, I'm not sure exactly what the question is here, but the questioner is saying, and this is Charles, is saying, do you mean in relation to complexity of cancer? I don't know when I said that in the talk, but none of this is simple. So I could have actually said that about cancer, obviously, in terms of its complexity. But I will also say that solving this problem is not at all simple. I've never said it's simple, actually. It's, as I say, if I look at those 6 parameters, they're all quite complex, but fixable. So I think it's complex at every level, Charles, and that doesn't make it daunting. It just makes it complex. And my whole admonition on complexity is embrace complexity, because if you don't, and if you can't, then we're not going to advance from this field, in this field or a lot of other areas surrounding cancer. Mike asks, for the FDA to expend energy to provide guidance to the biomarker development community, work is required. A very, very dirty word and concept to all government employees. Well, okay, Mike. I was a government employee and worked very hard, and there are lots and lots and lots of hard-working government employees. I don't think that's going to be the issue. And if you think about actually the issues of what it's going to take to regulate the numbers of biomarkers that are coming in, the number of drugs that are coming in, especially now that that FDA has proposed regulating the laboratory-developed tests. And let me just spend a second on that. The laboratory-developed tests have been around for a long time. They have all kinds of names, and they're being used routinely in lots of ways. But understand that this laboratory-developed test story has stymied the development of the molecular diagnostics industry. You can go through the presentation and the submission of a PMA, and you can actually present it to the FDA, and you can have your PMA approved, and you may need to sell your test, and rightly so, for, I don't know, say it's $1,000. The next day, someone can come out with a laboratory-developed test and sell it for whatever they choose. Maybe it's $10 or $20 or $100. So it really undermines the business model. We don't talk about that in the regulation of laboratory-developed tests, and maybe we shouldn't, but it's a big deal in terms of this entire field. So I think you're right. It's going to take a lot of work on the part of the FDA. But I think the FDA is up to it. I think they have the personnel. I think they're ready to do it, and they've given themselves enough time to do it, and I think they'll do it very, very well. So laboratory developed tests, by the way, there is some concern too about, and so many of our biomarkers are being used as laboratory developed tests. There's nothing, particularly wrong about that, if in fact they follow the big six and they're really robust and they're measuring what you think they're measuring. So the other issue, though, is that laboratory developed tests, obviously, if they're done in one laboratory, not reproduced in another laboratory, and not actually being challenged anywhere, they may or may not be measuring what you think they're going to measure. And so FDA, in its wisdom, I think, is saying we need to know the test is reproducible. We need to know that the data are robust, and especially if it's a high-risk situation. So, let me answer the next question. I spent a little time on laboratory-developed tests because this is a very confusing issue, and one that will be sorted out, I believe, in probably the next 5 to 10 years. But as I said when we went through the slides, there's not a lot to concern those people who have laboratory-developed tests because they're going into a queue, and it's going to take some time for FDA to actually sort out the risk factors here. But if you have a high-risk test, you are going to have to go through a PMA, would be my guess. So have to get ready for that. So we have a question from Prakash. And it says, are these standards proprietary or open, and how do we access standards? So it's an interesting question. If I look across the modules that I showed you that the NBDA has come up with, and maybe I can just go back to that. There, I'm sorry. If I look at these modules, there aren't any standards. There are no standards. I mean, when you're deciding to go from here to here, there are no standards to tell you that you have essentially designed, for example, an experiment that's going to answer the clinical question. And when you get to translatable discovery, there's very little to tell you what standards you have to have in terms of what's it going to take to scale this up. I mean, you may have a test that works great in 5 samples or 10 samples, but if you want to scale this up, is it scalable? You have no way of knowing that unless you have some way of standardizing what it's going to take to scale your test up. We have no real standards for antibodies to speak of, so we don't have very many good antibodies. As you know, commercially available antibodies are often not very good. So in answer to Prakash's question, we have almost no standards. And when you get out here to qualifying your biomarker, standards start to be important, and certainly when you get into clinical trials, there are lots of standards. But if you don't have any standards and you get out here and you have to comply with standards, your test is probably not going to be sufficiently robust to get through biomarker qualification, which is now an official process with the FDA. And you should go online to FDA's website and look up the qualification process. Very interesting process. It's been a very slow series of developments, but it's actually, we are qualifying. I saw an announcement yesterday of a biomarker having qualified through this process. Mostly this process is skipped and we go right over here. But before I leave this, what we're trying to do at NBDA is to get the communities to come together to create these standards, and then to partner with places like the College of American Pathologists and ASCO and AACR and some of the laboratory, especially some of the laboratory associations that actually can implement these at the patient level. So we do lots of things at our laboratories that never get translated to patients. So we want to bring this to patients in a way that it's going to be broadly translatable across all the communities, community hospitals especially. That was a great question. So we have a question from Deborah. And so it says that we've heard about CDISC. Believe me, we've heard a lot about CDISC. So Ken Buto, who's here with me at ASU, actually helped to, when I was at NCI, we helped to get CDISC underway. So these are existing standards to support the acquisition, exchange, submission, and archiving of clinical research data. And there is enormous synergy with this, and I would say that CDISC, in my opinion, represents the future. There is an example of groups taking the lead, and these were initially government agencies, but it's now much broader. And the whole issue of data, which we did not talk much about in this talk, but as you know, big data is one of the banes of our existence right now. It's a big plus in that we're getting enormous amounts of molecular data, but it's also a major negative in the sense that we don't have the analytics and we don't have the standards for the data. CDISC is one of those, I think, very, very important breakthrough kind of community activities where people are working together very, very closely to make sure that once you get into the clinic, that the data that you're submitting is of high quality and you do have data standards. And I would argue that we should have CDISC-like kinds of activities all the way along this pipeline. And what drives this green arrow is data. So in early discovery, once you start collecting data, if that data is not sufficiently robust that it's going to take you all the way over here, so when CDISC kicks in for clinical trials, then you've got a problem. And so I really applaud CDISC for all the work that they have done and will do. And our goal is to make sure that we have standards that allow us to go from discovery to a translatable discovery or a technology and move it through assay development and performance in a way that it qualifies and can meet CDISC performance measures. So we have a question from Carmen, and good question. She's asking, is there any relationship with the FDA, with the NBDA Alliance and the Biomarker Cluster Alliance in Europe? And are you aware of the programs on translation into product under the EU programs? And are we indeed ready for having a product since there are no other standards? First of all, yes, we're very aware of the Biomarker Cluster Alliance. As I say, we launched the NBDA in January. So we spent 18 months in due diligence before we launched in January. So we'd spent a lot of time understanding the problems. We are just now setting up our relationships, and we hope that we can partner with the Biomarker Cluster. We know of their work, and we're very, very supportive of what they're doing. And then we're very aware of what's going on in the EU in terms of many of the programs. Many of them are actually, I think, quite innovative and quite exciting, and we, in many ways, are looking to Europe. Some of the biomarker work that's going on in Europe is along the lines that we have laid out for NBDA, and so we're very supportive. I think in many ways Europe has appreciated and understood this need for standards well before anybody else did. I don't think there are any really clear standards. Let me give you one example. When I was at the National Cancer Institute, Dr. Carolyn Compton, who actually is with me here and is our Chief Medical Officer now for the National Biomarker Development Alliance, created over about a 5-year period best practices for biospecimen collection, and those are up on NCI's website. As a national force or as a community, we have never adopted those standards, and my fear is that the government will actually come in and implement those standards, and I don't mean necessarily NIH, but this could become an item for Congress in terms of the whole issue of the Affordable Care Act, trying to get the best care to patients. Patient samples are critical. Patient samples are the stuff of personalized medicine. So we really need people to adopt the standards for tissue collection and stewardship and all the things that are really matter about a patient's sample. The worst thing that can happen is a patient gives their sample and they find out the sample is lost or it's mismanaged, and so they don't get the benefit of actually having given their sample in the first place. So I want to stop and end on time because I know everybody has incredible schedules. I want to thank all of you for being online today. I want to encourage you to embrace this opportunity. If this field can take advantage of solving these very fixable problems, then I believe that cancer research will receive a lot more positive attention, a lot more funding, not just from the government, but from private foundations and others, so that the science will get better and better, and we can deliver all of those discoveries to patients. There is no reason for us not to do this, none. And through the National Biomarker Development Alliance, I hope we can pull this off with your help. Your organization is free to join us, and as I say, if you go online in about a week, you'll see how to join us online. So thank you very much for joining us this morning, and go forth and change the world. Thank you.